Thanks a lot. It's an honor and also a pleasure to be here and to see some friends in the audience. And indeed, I will tell you about nuclear volume regulation in cells. Why does that way go forwards? Okay. So, exactly. This is a problem that's been known for over 100 years and has been observed in cells in interphase, in resting phase. And in many, many systems, the nuclear volume and the cell volume are proportional. And the work I'm going to tell you about today was part of the PhD of a very unusual student, excellent student, Dan DeViri, who had first degrees in chemistry, physics, and biology. And only somebody like that, I think, would have attacked this kind of problem. And he was so good that after he did his PhD, he decided he's starting his own company to take CO2 out of the atmosphere using the oceans. But he still works with us very part-time and is still interested in science. And I hope that after he solves the problem of CO2, he will come back to the pure science world. Some of the work that I'm going to show you about chromatin is the work of other people in my group, and I'll refer to these experiments done at Weizmann. This is data from this paper, and the cell volume is varying many, many factors, and the nuclear volume is more or less proportional to that, but we're not trying to get exact proportionality. As Jean-Francois, we always have in parentheses bruit or noise. So you're looking at ensembles of cells, et cetera. So we're not trying to get values that are characteristic of things like critical phenomena, but this goes over a range of many cell types and many cell volumes. And I'll give you the bottom line first. And the bottom line answer, after I go through several possibilities, and you'll see at the very end of the talk why I'm going through possibilities that are not the bottom line answer. Bottom line answer is that there's some osmotic equilibrium between the nucleus and the cell. And you have small proteins or ions that can easily pass between the nucleus of the cell. Those are the little green ones. But you have blue proteins that are mostly localized to the nucleus and red proteins that are mostly localized to the cell, and we'll understand why. It's due to active transport. And the difference in that osmotic pressure seems to control the proportionality of the cell volume and the nuclear volume. How that works out, what are the estimates and orders of magnitude is what I'm going to talk about. And as I said at the end of my talk, there seemed to be a tiny explosion of interest in this in the biological community, because I found 3 papers or 4 papers only in 2024 that started exploring this. And you'll see that people are starting to explore extreme conditions for cells and seeing that the simple osmotic equilibrium may not be the answer to everything. All right, so nuclear size regulation is important because it's interesting, because it a priori in simple systems seems to be proportional to the cell size, but the nuclear volume is important because it has to do with the regulation of where the chromatin is sitting in the cell and with gene expression. And in this biology paper of Levy, he talks even about pathologies in which the nuclear morphology is altered, most notably cancer, and it's unclear what changes the nuclear morphology and nuclear volume. So the first order of business is to try to understand the nuclear volume in wild-type cells, and that is what we're going to compare with experiments. But at the end, as I told you, we'll talk about extreme conditions. All right, so here is a simple example of one of the recent papers, actually, of a change of 50 in the cell size and the nuclear fraction of the cell volume is almost constant. In this middle one, it's going up a bit, but there's certainly an indication that the nuclear volume and the cell volume are almost proportional, because here you see the cell size, here you see the cell size for this kind of a salamander, and the volume goes like the cube of the size, and that varies by a factor of 50, yet the ratio of the nuclear volume and the cell volume are about constants. And the other interesting thing here is that the genome size is varying tremendously here by 2 orders of magnitude, and still the ratio of the nuclear size and the cell size is just about the same. So that's a first indication that maybe it's not just the genome size that changes the physics of that relationship. Before we go ahead, I have to say a few words about chromatin, just so we're all on the same page, because chromatin is how the DNA is packaged in eukaryotes, and that's what we're going to be talking about. We're talking about lizards, salamanders, frogs, humans, and also yeast. As most of you probably know, the DNA in the nucleus is packaged in the form of chromatin, in the form of sort of beads on a string. The DNA here is a green string, and these red beads are oppositely charged histones. So DNA in solution is negatively charged. The histones are oppositely charged. The DNA winds around the histones. And it's estimated that the negative charge of the DNA is cut down to about 50% because of the association of the DNA and the histones. But there's something else that happens now. You have now what you might in physics call a polyamphalite. You have a polymer with both positively charged and negatively charged regions. So the fact that you have these positively charged histones and negatively charged DNA is not only that the DNA will wind around the oppositely charged protein histones, but also that a DNA from over here might interact attractively with a histone from over here. So the chromatin can experience associative or attractive interactions due to this electrostatic effect. And we'll see more about that later. And this idea was first introduced in a paper by Rosen. OK, so now we know that DNA is in the nucleus. In eukaryotes, it's in the nucleus packaged in a very particular manner with these histone proteins. And I promise you we'll get back to that later. The way we started on this problem was that my student Don read this paper, interesting paper by Cantwell and Nurse, and it was published it's a review and published in a biology review journal. Nurse is a Nobel Prize-winning biologist, and that paper exactly asked the question, what determines the ratio of the nuclear volume to cell volume? We saw that's not only intellectually interesting, but it's also interesting because changes in nuclear volume can give rise to various disease states. And what they did in that paper was list and explain very nicely several possible ideas. And some of them they shot down. They said probably not relevant, and others they said are probably relevant. After Don read that, he showed it to me, and he said, you know, this paper talks about all the right things, but there's not one word there about mechanical equilibrium, about osmotic pressure, about interfacial tension. There's no translation of the biological words into physical concepts. And what we did in the paper that was eventually published was do that translation in the simplest way possible. So this will be sort of an easy talk to follow, because everything are simple back-of-the-envelope estimates that you can make, at least if you know some polymer physics and soft matter physics. And then we want to see which of the effects that Cantwell and Nurse talked about, which of them are important. So they have a whole list of effects. And the first one that they talked about was genome size. And they quoted previous papers that called it nucleoskeletal theory, that the genome, or the chromatin, is like a skeleton in the nucleus, and it's what determines the nuclear size. And we can translate that into physics by saying that there are two relevant effects if we want to think of that. The first one is the osmotic pressure of the chromatin as a polymer. So if you think of the nucleus as a spherical ball filled mostly with water, chromatin only occupies, depending on estimates, between a few to 20, 30% of the nuclear volume. That polymer, if it's uniformly distributed in the nucleus, and if you say that water is a good solvent for chromatin, then it will bang around, it'll fluctuate, and it'll exert a pressure on the nucleus. And of course, the cytoplasm has its own pressure. You have ions, you have ions that can go back and forth, but you have proteins in the cytoplasm. You might have elasticity in the cytoplasm. So there are two pressures, one from the nucleus, one from the cytoplasm, and their balance is going to give you the nuclear volume. The other effect that has to do with the nucleus is the fact that the chromatin is charged. It's not as charged as DNA, because you have the positively charged histones, but it is still charged. It's charged at about half the negative charge of DNA. And that charge gives rise to counterions in solution. Of course, the nucleus is going to be neutral, so the fact that the polymer backbone is charged means you have counterions in solution that have gone solution basically because of entropy, but there are electrostatic screening effects involved as well. And those ions themselves exert a pressure on the nucleus. So we said, let's try to get a back-of-the-envelope estimate for these two effects, the polymer pressure and also the counterion pressure. And let's see if there's sort of the dominant effects. And this is also something that Cantwell and Nurse said, let's look not at the physics, but at the biology literature. And their conclusion was that the nuclear skeletal theory did not explain the nuclear size. And they basically gave many, many references to papers where people changed the genome size and they didn't see significant changes in the nucleus. And that was our conclusion as well, but for a physics reason. Okay, the next thing they talked about was cytoskeletal stress coupled to the nucleus. So you not only have pressure in the nucleus, you have pressure outside, and perhaps the cytoskeleton or some actin network within the cytoplasm of the cell is the source of the pressure on the nucleus. So the first one is pressure in the nucleus, second one pressure on the nucleus. And we made some estimates of that based on experiment. That doesn't seem to be the explanation. The third one, they gave a more fancier name and basically has to do with nucleoplasmic transport. And this is the one that I'll try to convince you seems to explain the order of magnitude for the regulation of the nuclear size relative to the cell. And I'll show you an experiment about that in the end. And this means that molecules cannot transfer at will from the nucleus to the cytoplasm, because the way they get out of the nucleus, which is surrounded by an inner and an outer nuclear membrane, is through the nuclear pore complex, a very intricate complex of proteins that allows small molecules to diffuse back and forth, but large molecules cannot unless they're attached to a nuclear localization signal, and that involves active transport of ATP or GTP and how different molecules get in and out of the nucleus. And this, we believe, which has to finally do with the concentration of proteins in the nucleus and the concentration of proteins not allowed in the nucleus because they don't have this correct transport, that gives you different protein concentrations in the cytoplasm nucleus. And the order of magnitude of that seems to be larger than these other effects. So we're just following this biology paper, but saying, can we translate this into physics or into chemical physics or polymer physics ideas? Okay, so just to calibrate us, I did the exercise of saying if we have a dilute solution of molecules, ions or larger molecules, proteins, which will be important in our case, what would be the osmotic pressure if the solution's dilute? And you probably know the ideal gas law, that the pressure is proportional to the concentration, to the density of an ideal gas. When you have molecules in solution, you don't talk about density, but you talk about concentration C. And for a concentration C of micromolar, the pressure is about 1 to 2 pascals. For millimolar, it's 2 kilopascals, and for molar, you have 2 megapascals. And we'll get back to this later. Okay, I think I'll skip this for the time being. Okay, so first I want to just convince you that even if you have elastic forces, let's say through a cytoskeleton or actin network, or maybe through the chromatin network, as we'll see, that the pressures due to small osmolytes, to ions, and also to amino acids, and also to proteins, is quite substantial. And this is unrelated to the question of nuclear volume, but is a measurement of Ming Gao and David Waits of the modulus of the cell in pascals for various cell volumes. And what I'm showing you here is the bulk modulus of the cell, where you just press on it. It's an osmotic modulus. You're not squeezing any molecules, but water can go out of the cell and molecules can go, small molecules, ions can go in and out of the cell in a way that could be complex, but we're not going to talk about that now. And in the end, they get megapascals. And that is in line with the fact that you have about 200 millimolar of various small ions and small molecules in the cell, and that's consistent with what I showed you before. So when you have a system and you have a coordinated network that is putting force on the system, exerting pressure. But you also have molecules that can go in and out, and they exert pressure if they're going to be localized someplace. Then the small molecule terms are often the most important. Okay, so now let's go talk about nucleoskeletal theory. The first thing that everybody thinks about is the nuclear size has to do with the polymer banging into the nucleus, diffusing within the nuclear volume and exerting a pressure. The cytoplasm has its own pressure, and the balance of the two is going to determine the nuclear size. So this was called nucleoskeletal theory, and was introduced in 1978. And the assumption here is that chromatin in water, right, is a polymer in water, is a good polymer and a good solvent. So that is a very strong assumption, and we'll see that there's some experimental evidence that just by chance happened to be done at the Weizmann Institute by my collaborator Talila Volk that puts into question whether in live cells that's really the case. But we said, let's just go with this for a moment, and let's assume that the chromatin's in a good solvent, and let's see what an upper bound on the pressure would be. So let the chromatin just go as a random polymer in the water, and there's a very beautiful paper by Elie Raphael, and also some other papers that have looked at how a polymer in a confined environment of the nucleus, how the polymer exerts pressure on the nucleus. And they've just looked at a polymer with excluded volume. The exponents here are the Flory exponents, where the radius of gyration will go like the number of persistence lengths in the polymer to the 3/5. So it's a simple-minded estimate. So here you have a polymer in between two flat plates. Here it's in a cylinder, and here it's in a spherical, we'll call it the nucleus. And your polymer number of persistence lengths is n, and you want to know how the pressure within the nucleus varies as a function of n. So over here, your polymer is really organized in a series of blobs. Within the blob, the polymer doesn't really see the confinement, and it's behaving like an excluded volume polymer. So the radius of a blob goes like the number of persistence lengths in the blob to the 3/5. but at bigger length scales, it sees the confinement, and that confinement restricts the entropy of how you're going to arrange all these blobs. So every time a blob has a collision with a wall or another blob, you lose a kT of entropy. And that's famous blob scaling theory that I believe that Degennes introduced. And if you're interested, there's not so many papers making systematics of this, but somewhat systematics were made in a review paper by Avi Halper many years ago. Anyway, if you confine the polymer over here, it's pretty easy to convince yourself that if you make the polymer longer, you'll just get more blobs. And the free energy goes linear in the length of the polymer. However, over here, you have to worry about the fact that if you're going to change the volume of your confinement, your blobs are going to get more concentrated, and then that's going to cost you entropy within the blob. And with a very, very simple scaling argument that you say that the pressure will be invariant if you change the length of the polymer by a factor lambda and the volume by that same factor lambda. So we're going to have an invariant system if we magnify the polymer, but also change the volume. Raphael and coworkers showed that the free energy goes like n to the 9/4, and the pressure is derivative free energy with respect to volume. It also goes like n to the 9/4. So we have some scaling estimates of how the free energy, and hence pressure, which is the negative of the derivative of the free energy with volume, should scale. And this is the formula you just write down, taking that free energy and taking its derivative. Why you get 9/4, it has to do with the fact that the radius of gyration within a blob goes like the number of persistence lengths within the blob to the 3/5. But the fact that if you just multiply up the polymer, magnify the polymer by a factor and magnify the volume by the same factor, the free energy shouldn't change. It's a simple argument. I won't do the algebra. And you can put some numbers here for both humans and yeast. And for humans, you see that this would give you a pressure of about 30 pascals, and for yeast, something much smaller. And probably the other contributions we'll talk about, namely the contribution of proteins that are localized to the nucleus versus proteins that are localized to the cytoplasm because of active transfer, those turn out to be, in our simple estimates, much larger. So we wrote other contributions may dominate. It's very hard to say what is the story here, but other contributions seem to be bigger by order of magnitude. So this nucleoskeletal theory sounds very nice, but the pressure is indeed very small. Now you could say, all right, but this is a polyelectrolyte. It's a charged polymer, as I said, with both positive and negative parts. Sorry, before I get to that, I wanted to say there are two arguments against the nucleoskeletal theory that are qualitative arguments, not just our order of magnitude arguments. And one of them brought in the paper by Nurse and Cantwell, works Newman and Nurse, is that changes of DNA content did not change the nuclear volume. And they looked at changes of orders of factors. At the end, I'm going to show you changes that are larger by some workers just this year where they question this. But I did show you the salamanders where there was 2 order of magnitude change in the genome, and the nuclear to cell volume fraction were the same. The cells got bigger, of course. But the other reason has to do with the work that I hinted at before, is questioning whether chromatin in the nucleus is a polymer in a good solvent that wants to take up as much volume as possible because of entropy, or a polymer in a poor solvent. A polymer in a poor solvent means that the polymer segments are attracted to each other more than they are attracted to the water, to the solvent, and the polymer then will collapse. It'll attract. And the group of Talila Volk, 2021, published a paper with some theory by my group, with pictures of, microscope pictures, these are not pictures, but microscope pictures of the nucleus of live Drosophila nuclei in muscle cells, but they looked at other cells as well. And one of the important differences that they didn't take cells out and spread them on a slide. When you do that, you can dehydrate the cell, and Waits and Go showed that. They also didn't fix the chromatin by putting in various chemicals like formaldehyde that fix the DNA and chromatin change. These were actually on live fly larvae, so baby flies, and they know they're alive because they have them in a certain holder, and the flies can move their muscles, and they're looking at the muscle cells. So their pictures showed very dramatically that in the wild type, the chromatin, which is red here, they stained for a certain histone that's found in both euchromatin and heterochromatin, is phase separated from the aqueous phase. And these lines here, these circular lines are not from the microscope picture. They're the way that they discretize the volume to actually quantify them. Now, the chromatin in the wild type is also attracted to the nuclear envelope for reasons I won't go into, but there are lamin adhesive domains of DNA. And as you see, it's phase separated, but it's phase separated on the outside, on the periphery. That means it actually has a pretty big interface with the water. And you say, why would it want to do that? Because the water, if chromatin is self-attractive, the water-chromatin interaction is repulsive because the chromatin phase separates. Why would it make such a big interface? And the answer is, well, it's attracted to this green outer nuclear envelope. Indeed, if they do various mutations to reduce this attraction dramatically, they still see that the chromatin is phase separated. It's self-attractive, but now it's in the center. Why is it in the center? Because if it no longer has a molecular reason to be attracted to the nuclear envelope, then it's going to make a smaller interface with the aqueous phase, which is what a system will do when it is self-attractive. It wants to have the smallest interface possible with the phase of molecules that it's not attracted to. And this only happens because of this. But in both cases, the chromatin is phase separated. Now, once Talila, when we published this paper, we started hearing from lots of people, especially Shiva Shankar, saying, oh, we saw this here, and we saw this here, and we saw this here, and we saw this here. And it's true, and sometimes it was published, but it was never investigated systematically and quantitatively as these guys did. And moreover, it wasn't thought about in terms of polymer in a poor solvent and phase separation. But if this is correct and generalizable, and it's also been observed in human immune cells, that it's in the same paper in Science Advances, and salivary gland cells of the fly, and these were muscle cells. If it's general in live cells, then it means that in the live cells, at least, the DNA, the chromatin, is not a polymer in a good solvent, but is phase separated, and therefore this picture of the nucleoskeletal theory that assumes that the chromin wants to take up as much volume as possible is not applicable. All right, so here again to remind you what is chromatin. I think I've said enough about that, but I want to make sure. And actually, this is just reviewing what I said. I forgot I had these slides. So in a good solvent, the chromatin just does a random walk. It wants to occupy as much volume as possible. And in a poor solvent, the chromatin will collapse. And the reason why chromatin is attracted molecularly is not yet resolved. There are several workers that have worked on this even in vitro. The groups of Hensel and also the group of Rosen. And it's not quite clear. Rosen suggests that it is the attractions of the negative DNA linkers, these linkers here, to the positive histones, that makes a linker from here be attracted to a histone and condenses chromatin. But there are many other proteins involved in actual in vivo systems, so that's not clear. However, Hensel and also Rosen, they have in vitro studies of short pieces, constructs of chromatin that also collapse. And this is what I showed you. So we'll go ahead now to the next possibility. Yeah.
It's a very naive question, but as far as I know, the DNA can be condensed and decondensed in terms of what biologists refer to that, and in the nucleus it's supposed to be decondensed as compared to the...
Okay. If you have DNA in a bacteria where you have no chromatin, okay, you might have some other things, but naively the DNA is just DNA and not chromatin. Then if you have various ions, usually multivalent ions, and people have done this in vitro, you can condense the DNA. That's condensation of DNA, and these positively charged ions form sort of bridges between the DNA. In chromatin, at least according to this, we didn't work this out molecularly, but in chromatin, the idea is that you don't need the multivalent ions because you have multivalent positively charged histones. So it's related to what you said.
What I meant is that so chromatin can be condensed.
Chromatin is condensed. Yes.
But it can also be decondensed. I guess usually it would be referred
well, in when cells divide, the chromatin that the chromosomes that in high school they draw sort of like something like this. This is very, very condensed chromatin. So this is more condensed than in the live system they're showing you. Okay, so we've shown that the nucleoskeletal pressure is on the small side compared to some of the others we'll see. And we've also seen that the chromatin itself might not be taking up the entire volume of the nucleus. It may be separated from that volume. So let's look at the next nuclear effect that has to do with chromatin, which is the osmotic pressure of the ions that are neutralizing the positive and charges of the histones and the negative charges of DNA. You have ions in solution, and they exert an osmotic pressure in the nucleus. And it's easy to estimate that. There is a simple theory called the Donin equilibrium, which says that if you have an equilibrium system, you have to have the same chemical potential for the ions inside your system and outside your system. So if we imagine the nucleus and the cytoplasm, and we think of ions in the nucleus, those ions are free to travel through the nuclear pore complex. They don't need active transport to get out of the nucleus. They're small enough to fit in the nuclear pores. And they're in the cytoplasm, they're in the nucleus, they're in presumably chemical equilibrium. And you can use these Donin ideas, which says that the ion should have the same chemical potential inside and outside, and that the nucleus should be overall neutral. So why is the nucleus different from the cytoplasm? Because it has the chromatin, and the chromatin is charged. And the chromatin is localized to the nucleus. It can't get out under normal conditions to the cytoplasm. So you have this big chunk of negative charge that is fixed to the nucleus, and you have positive and negative ions that can go back and forth. And you have the two conditions, that the chemical potential of the ions, which is basically the log of their concentration, should be the same in equilibrium, and that the nucleus should be neutral. And when you do that, you can work out the pressure difference between the ions in the cytoplasm and the ions plus chromatin in the nucleus. And you can work out a very simple formula for that. It goes quadratically like the charge concentration of the chromatin. So N sub C is the charge concentration of the chromatin, not the number of molecules per unit volume, but the number of charges per unit volume of the chromatin. And NS is this naive salt concentration. So you have screening ions, you have ions that are screening in addition to the counter ions of the chromatin. You have many other ions in the cell, both in the cytoplasm and nucleus. If those ions can reach chemical equilibrium, because they can diffuse in and out of the nucleus, they will screen the charges in the nucleus along with the counter ions of the chromatin, and you could work that out. And it goes quadratically with the chromatin charge. And again, you could work out these very, very simple estimates, and you find, well, the numbers get bigger. You have more pascals than you had for the chromatin itself causing the pressure. The ions here give you a larger pressure, but it seems that the other forces that I talked about, namely the proteins that are localized to the nucleus versus the proteins that are localized to the cytoplasm, can give you a bigger pressure difference. Also, this will be reduced by chromatin self-attraction, but I'll point out that if we do, as I said, extreme mechanics to the nucleus and greatly change things, that the pressure goes quadratically with the charge of the chromatin. So if we greatly change the charge of the chromatin, we may get to pressures larger than this pressure for human cells, wild type, as they are not manipulated. Incidentally, the same thing goes for the osmotic pressure of the chromatin as a polymer. I pointed out that that pressure scales like the number of persistence lengths to the 9/4. So the numbers that I showed you that were small were for estimates for chromatin as is in the human nucleus and in the yeast. But that 9/4 power means that if we'll increase the genome greatly, those effects can start to become important. But for the numbers that we have, which are the wild type of human and wild type of yeast, these seem to be small effects. Elastic forces of the cytoskeleton. So now we'll move from the nucleus to the cytoplasm, which could have the actin cytoskeleton. The actin cytoskeleton in the cytoplasm is organized in two ways. One of it is the cortex, which is an actin layer that's not very, very thick, that is connected to the membrane of the cell. And that is populated by high concentration of actin. But you also can have a filamentous actin network pervading the volume of the nucleus. And there were estimates of these moduli, again, by Go and Waits. These are the shear moduli that they estimated by putting in various kinds of magnetic tweezers and shearing the cell. So first they can shear the cell from the outside, measure the bulk modulus, and it can go inside and get an estimate of the cytoplasmic modulus. You see the cortical modulus on the order of 10 kilopascals. 10 kilopascals is always the modulus or the pressure or free energy per unit volume for biopolymers. All right? But the cytoplasmic modulus is much lower because the cytoplasm might have a filamentous actin network, but it's much, much less dense than the cortical actin network. So we said we think even in the cytoplasm, other forces may dominate. So now let's get to the proteins that I already told you will be the important factor, the way that we're thinking. And we're going to look at the osmotic pressure of cytoplasmic proteins and osmotic pressure of nuclear proteins. Now, if the nucleus and the cytoplasm were in thermal or chemical equilibrium, and proteins could go in and out of the nucleus, then there's no reason for their concentrations to be different in the nucleus and the cytoplasm if they can reach chemical equilibrium, except maybe by some boundary conditions. So what does it mean to say osmotic pressure of cytoplasmic proteins and of nuclear proteins? If you say proteins are localized to the nucleus and cytoplasm, what you're saying is that those proteins cannot diffuse at will between the nucleus and cytoplasm. And that indeed is the case. This is a sketch. Here's the nucleus, and here's chromatin. And there's a lot of stuff in here that's not relevant. There's a nuclear envelope. Now, it's complex. It's formed by proteins in an elastic network called the lamina, but it also has two bilayers of mostly lipid molecules and some other proteins. But the point is that a large protein can't really get out of this mesh, especially with the bilayers over here. But there are the nuclear pore complex. A complex, you want to think of them as pores, or they're not holes because they're surrounded by proteins, but they're ways that molecules can get in and out of the nucleus, and that, of course, is very important. We want to get to the ribosomes, or we've got to get the RNA out, and other molecules have to come into the nucleus. But this pore complex, and the numbers are here, so the pores are rather big, but they're not as big as 60 nanometers, you might say, because they're formed of these brushes of polymers that reduce the effective pore size. And molecules require a signaling molecule, a nuclear localization signal, to piggyback upon and to go into the nucleus. The biochemistry of these nuclear localization signals is not trivial. And my colleague at Weizmann, Michael Elbaum, has looked at this for many years. But it means that the transport of proteins, not small ions, from here to out here to the cytoplasm requires some activity, requires ATP or GTP. Well, here's again the picture in our head. So we have the blue proteins are preferentially transported into the nucleus, the red ones preferentially transported out of the nucleus, and the little green ones are small proteins or amino acids or molecules that can just go and freely diffuse, and we'll reach Donin type of equilibrium. The chemical potentials will presumably be the same. But these big proteins need a process involving energy, involving ATP or GTP, to get in and out of the nucleus. So we cannot assume that they're at chemical equilibrium. And the easiest way to think about that is about an active transport. When you have diffusion, you have equal probability of going in as going out. But when you have active transport, that detailed balance, which is what leads to the equal probabilities, means that your probability of being transported in is not the same as the probability of being transported out. Why? Because your localization signal may detach, may lose a phosphorus in the nucleus. There are a lot of active processes that make this a non-equilibrium process. I'll give you a reference in a minute, I think. So in equilibrium, what you would expect, well, in general, in steady state, what you expect is that the flux of molecules, the number of molecules per unit area per unit time, which is the rate of transport that molecule into the nucleus from the cytoplasm, should be equal to the rate of transporting that molecule from the nucleus to the cytoplasm. If the fluxes are not the same, you're going to have an accumulation of molecules in either the nucleus or cytoplasm. So steady state means that the fluxes are the same, not the concentrations, but the flux is the number per unit volume per unit area per unit time, which involves the transport coefficients. If the two transport coefficients are the same, the concentrations are the same, and system equilibrates. But if they're not the same, because you go in and you get a phosphorus attached to the transporter, or other things may happen, these two transport coefficients are not the same. And this is the very simplest way of understanding that some proteins may be transported preferentially into the nucleus. They'll have a high rate of going into the nucleus and a low rate of going out. If it were diffusion, they could go in and out with equal probabilities. And other proteins may have a high rate of being transported out of the nucleus, but a very small rate of being transported into the nucleus, and those proteins will preferentially be in the cytoplasm. And this effectively localizes certain proteins to the cytoplasm or nucleus. So it's activity, breaking of detailed balance, the fact that the transport rates in and out of the nucleus are not the same. It says you have a different concentration of certain proteins outside and certain proteins inside, not the same protein. We have many protein types. And a little more is written in our paper about this. All right, this is just a reference to the work of Copito and Elbaum, who many years before looked at this problem of transport into the nucleus and looked at which aspects are equilibrium and which aspects are non-equilibrium. And the picture is complicated. It involves a molecule called RAN-GTP and how it attaches to the localization signal, but they came to the same conclusion. And in this paper, it seems that most proteins are localized either to the nucleus or the cytoskeleton, to the cytoplasm. I mean, extreme example are histones that are mostly in the nucleus because they're grabbed onto by the DNA. Well, with this, we can estimate the osmotic pressure, because if the proteins are more or less equally balanced in the nucleus and cytoplasm, you can estimate how many proteins there are in the cell. You know their concentration, and you know what the osmotic pressure will be. It will be Kt times the concentration of the localized proteins. And when you do an estimate of that, it's not very exact, but it turns out to be in the kilopascal range. Doesn't have to do with elasticity, has to do with the fact that the osmotic pressure of these localized proteins in the nucleus is not the same as the localized proteins in the cytoplasm. And that seems to dominate the other effects: chromatin osmotic pressure, chromatin electrostatics, cytoplasmic elasticity due to the wispy actin network that's in the cytoplasm. This seems to be the dominant effect. And it doesn't take much to say that if we will have a nucleus that is floppy, and I'll show you some experiments on floppy nuclei, where the nuclei is not taut, it's not blown up like a balloon. If we can, in those cases, neglect the interfacial tension of the nucleus, which might only be entropic, that's an approximation, then we're going to have an osmotic pressure of these localized proteins in the cytoplasm, an osmotic pressure of these localized proteins in the nucleus, and mechanical equilibrium, not chemical equilibrium, not thermodynamic equilibrium, will be when these two pressures are equal. And the pressures by dilute solution theory is just proportional to the concentrations. And doing one more step of algebra, you can show that the cell to nuclear volume just goes by the ratio of the proteins localized to the cytoplasm divided by the proteins localized to the nucleus. So this idea that for each cell type, you can't predict a priori what these proteins, how many proteins will be localized to the nucleus and how many will be localized cytoplasm. But what you can predict is that that's going to be fixed for each cell type, and therefore the ratio of the cell to the nucleus should be, volumes should be constant. So that's our conclusion, and I'm going to, I have 10 minutes, I'm going to just show you a little bit of an experiment related to that, and then some things for all of us to think about. Okay, so here's a very nice experiment by Finnan et al. From a number of years ago. One of the people who participated in this is Kevin Chaloux, who you guys might know. And they looked at cells under hyperosmotic. That means the nuclear envelope is relaxed. It's pushed by the pressure outside of it. And hypoosmotic. When you are hypoosmotic conditions, the nuclei wants to expand, and the nuclear envelope will be taut. Then you're going to have a Laplace term in the pressure. Two pressures will not be equal, but the two pressures will be related by the tension of the nuclear envelope and the nuclear size. So Laplace law for a sphere is delta P is 2 gamma n over the nucleus size. So for a floppy nucleus, where we haven't inflated it, we can say gamma's small, and those were the estimates I've shown you before. But when the nucleus is taut, gamma is in some units, in 3D units, kilopascals, and then you can convert it to the gamma. It's an elastic network with an elasticity of kilopascals. So what you see here is that the circles are the cell volume, the squares are the nuclear volume, and they track each other pretty well under hyperosmotic conditions. Hyperosmotic is when you're pushing on the nucleus can be floppy. It's too much area. It has no tension. Once the nucleus gets inflated by hypoosmotic conditions, the nuclear envelope gets taut. It's like the second picture over there, and you lose this proportionality. So all the estimates that I showed you over here were for floppy nuclei. Why? Because we said all we're doing is equating the pressure of the cytoplasm and the nucleus. We sort of forgot about the Laplace law that if you inflate a balloon, the difference in pressure inside and outside is going to be related to the tension of the balloon surface. And the reason we forgot about it is because you can have conditions where the nuclei are fairly floppy, and then you just equate pressures. When they're not, it gets more complicated, and you have to know what this gamma is. But definitely what they saw is that the scaling of cell volume and nuclear volume scales when the nuclei are floppy, but not when you have completely inflated them. A more recent experiment that's more to the point of our work, which was, is that you have different proteins localized to the nucleus and different proteins localized to the cytoplasm. Why? Because they're not in thermodynamic equilibrium, because their transport is active. They don't have the same rates of going in and going out of the nucleus. Was in a paper that came out just about the same time as our paper, and we each learned about each other's papers only when we were reading the proofs, and then we were still able to put in a reference. And they were looking at yeast, and they modulated the osmotic buffer to change the sizes. And one of their conclusions, they found that Vn over Vc, the nuclear volume, cell volume, is fixed, and for every set of conditions. But what they did was they inhibited nuclear export. So various biochemical means, they plugged up, if you want to say, these nuclear pore complexes so that export from the nucleus would be inhibited. If export is inhibited, you have an accumulation of proteins inside the nucleus. You have more proteins in the nucleus, then they exert more pressure and the nucleus expands. So we were very happy. This was published the same year as our paper, and they were happy to see us, and we're happy to see them, and everybody was happy. But I'm not sure you guys could tell me if de Gennes said this, because I don't remember. I think he once said nature is wicked, but I couldn't find a reference, so I don't want to quote any. But biology is certainly wicked. That once you think you understand something in physics terms or chemical terms in biology, biology can find a way to get around your concept. And as I said, there are 4 papers that have been published just since January on this problem of nuclear to cell volume ratios. In the specialists, they call it N to C, nuclear cell volume ratio. And I'm going to talk about 2 of them. First, I'm going to just show you an earlier paper that I referenced before by Ming Goh, who's at MIT, and David Waits, who's at Harvard, where they looked at cells that were spreading on surfaces, and they could pattern the surface and change the size of the cell, or they could look dynamically as the cell spreads slowly. And as part of their long paper, they looked at the nuclear volume versus cell volume, and for all different conditions, it seemed to follow a linear path. That means that this ratio is constant. Again, what does a ratio constant mean? It means that more microscopically, the ratio of proteins, number of proteins in the nucleus and number or concentration in the cytoplasm is the same, even though the cell has spread differently. So that's very nice. But then there is a very, very recent paper, and one of the co-authors on this paper is Mathieu Piel, and their conclusion is that this isn't always true. Here is the mean NC. NC is nuclear to cell volume ratio as a function of time, and they're doing it as a function of time as their cell spreads, and they see that for a wild-type cell, it increases with time. It doesn't stay constant. Now, they didn't do it for many hours to see if it'll go back to a homeostatic value, but that is a very interesting finding. Of course, the theory that I told you about did not take into account adhesion. It did not take spreading. It was just a picture of a cell in suspension with its nucleus. So there's nothing contradicting what we did here. But this says that once you put forces on the cell, why is a cell spreading? Because there are forces that are pulling it out on the substrate. They act on the cell membrane, on the cell cortex, and there can be stress fibers that can even transmit these forces to the nucleus. Once you have these forces of spreading, then the NC ratio, the volume ratio, is not necessarily a constant. However, interestingly, when they treated their cells with a drug that arrests the cell cycle, they found that even though the cell spread, then the NC ratio remained constant. And these experiments, when you look back at the little words in the materials and methods that theorists hardly ever look at, but you have to look at the small font there, they tell you they synchronize the cells various ways to avoid going through the cell cycle. So there may be a difference into how forces, such as spreading forces, affect the nuclear to cell volume ratio when cells are wild type or when cells are treated and their cell cycles disrupted, or maybe the cell spends much more time in interface. So this is one of the recent papers, and here are two more. What we concluded was that the difference in pressure by these order of magnitude back of the envelope estimates comes from the difference in pressure of proteins localized to the cytoplasm versus proteins localized to the nucleus. But Levy and coworkers just published a paper showing that nuclear growth and import can be decoupled. So he does extreme mechanics. He does very, very large changes, biochemical changes to the nucleus, either with drugs or by, in an unnatural manner, packing them full of more chromatin, and says, oh, we hardly changed the import rate, but we saw that the nucleus grew. And he's very careful. He just says they can be decoupled. He doesn't say that in the wild type that the explanation that we offered and the yeast paper here, where they did a very systematic perturbation, that's not relevant. He just says, I can find conditions where the transport rates are not changing, but yet the nuclear size is changing dramatically. And in development, this is certainly true. And here's another paper just published again, this talking about early embryos, where again, the nuclear and cell sizes are not proportional. So I think it's great to feel at least that I have a somewhat friendly audience, so I can show you things that we have thought about and things that we have tried to estimate and tried to say that at least in the wild type, it's the osmotic pressure of these localized proteins that are important, but there's much more to think about and more experiments to do and probably more theory to do. What are the forces on the nucleus as the cell spreads? My friend Ram Adar, who was a postdoc with Jean-Francois and also worked with Pierre, has thought about forces on the cell and osmotic equilibrium, but not about forces on the nucleus. So we have to go back and think now about extreme mechanics, not just try to estimate these pressures in the wild type, but what happens under a range of conditions. And I remind you something I said at the beginning. The ion pressure increases with the square of the chromatin concentration. So if you increase the genome enough, you will start to see some effects of that. And the chromatin pressure itself increases as more than quadratically with the genome size. So what you conclude for wild type might not be true for extreme conditions, and I leave that as something for us to think about. Thank you.